Okay, so guys, no, our topic for today is about the nucleus. Okay, so next week we will be discussing naman no about the cell cytoplasm. So here in this topic, just like the topic we'll be having next week, okay, so cell cytoplasm, we'll just okay discuss no yung mga how do you call this one, the major okay components or structures of the cell nucleus. Ganun din sa ating cytoplasm and of course the function okay of these ultrastructures. So in this topic we will be uh, describing now okay the appearance of the cell nucleus, okay, its components. So what can you see in its cell membrane, okay, or what are the differences okay between your heterochromatin and your euchromatin, okay, and of course other structures okay that forms now the nucleus okay of the cell. So when we say nucleus, we all know okay that this is a struct or a specialized okay structure that of course occur in most cells, okay, except syempre sa mga bacteria natin, okay, and we all know that this nucleus okay is of course separated from the rest of the cell by a membrane that we call now your nuclear membrane, okay. And of course, the nuclear membrane is a membrane, okay, enclosing the nucleus that is continuous, okay, dun sa ating endoplasmic reticulum. So mamaya makikita nyo siya, okay, dun sa ating picture. Okay? So when we say nucleus, okay, I believe you have already discussed about the nucleus no sa ibang subject ninyo at maging ganun din sa mga lower years ninyo or in high school, for example, in biology. Okay? So when we say nucleus, of course, it contains a blueprint for all cell structures and activities. Okay? So ibig sabihin, this nucleus now controls, okay, and regulates the activities of the cell, like, for example, growth and metabolism. Okay? And of course, the nucleus carries the genes, okay, structures that contain now the hereditary information. Okay? So guys, no, sabi nga nyo dyan, it contains the molecular machinery to replicate its DNA and to synthesize an RNA. Okay? So ibig sabihin, siya yung central portion, okay, nung ating cell. And it's the one, okay, that provides now, okay, the information needed by the cell to perform, okay, its function. Okay? So guys, now we have here a structure or a picture of the cell nucleus. So ito, syempre guys, makikita nyo ito doon sa um, not central, okay? It's not always centrally located kasi there are cells na yung kanilang nuclei ay eccentric. Ibig sabihin nasa peripheral part or medyo gilid siya. Okay? There are those naman na nasa gitna. Okay? So, yeah. Ito siya. This is, hold on. Okay, this is the nucleus, okay? And this is actually the cell. Okay, so the cell and the rest of these outside the nucleus is of course what we call your cytoplasm. Okay, and separating now the nucleus dun sa ating cytoplasm is what we call as of course your nuclear membrane. Okay, so if you can see here, this is the whole of your nucleus, guys, no? Okay, and then it's actually continuous dun sa ating rough endoplasmic reticulum. Okay, so if you can see, it's rough because it has the attached ribosomes. Okay, ganun din guys, no, dun sa ating nuclear membrane. Okay, they are, the nuclear membrane, I should say, is actually rough due to the attached ribosomes. Okay, and of course, contains now numerous pores. And we call these your nuclear pores. Okay, and at the center of this nucleus is the structure called as your nucleolus. Okay? And here, kumbaga yung parang pinaka-cytoplasm, okay, nung iyong nucleus, parang counterpart ng cytoplasm mo, pero sa loob ng nuclear membrane is your nucleolema. Okay? So it contains now the chromatin materials dispersed, okay, dito sa ating paligid, okay, ng ating nucleolus. Other than those that are found dito sa mismong nucleolus natin. Okay? So, we will be talking about these structures, okay, in this topic. So, this is just a very short topic. Why? Because here we will not be discussing about cell cycle, meiosis, mitosis, because you'll be be discussing this, okay, topic sa ibang subject ninyo, okay, in cytogenetics. So guys, no, we have here a picture, okay, showing you now the structure of your nucleus under your electron microscope. Okay, so this is our picture A. So this is under your transmission electron microscope. So this is the appearance of a typical cell nuclei, okay, or nucleus. So if you can see here, it has electron dense heterochromatin. Okay, so yung mga dark areas natin guys dito, these are your heterochromatins. Okay, and of course, yung mga parang granules sa paligid ng heterochromatin mo, those naman are your euchromatin. So mamaya, we'll discuss what's the difference between your heterochromatin and that of your euchromatin. Okay? So the arrows indicate the nucleolus associated heterochromatin. So we have here, so mamaya i-discuss natin guys, no, ano ito? This is your nucleolus. So ano yung area dito? The pars fibrosa and the pars granulosa. So what are these, okay, areas in the nucleolus? Okay, what are their function? How are we going to describe them? Okay, so also we can see here, no, the presence of the nuclear envelope. So this one here, these arrows, okay, yung pinopoint ng mga arrows natin, this, okay, or this is your nuclear envelope. Okay, so of course, for us to better understand this, we have here a schematic representation of the cell nucleus, okay, based on that TEM image. Okay, so if you can see guys, no, yung sa nuclear envelope niya, this is the inner portion. Okay, and then this is naman the outer portion of the envelope or the nuclear membrane. Okay, and if you can see, the dark areas here, okay, ito yung mga heterochromatin ninyo. Okay? So, electron dense areas. Okay? And the less dense areas, okay, yung lighter portions, dito naman nakasuspend yung ating mga euchromatin. Okay? And if you can see, other than the inner membrane nung ating envelope, nuclear envelope, we have this outer membrane here, okay, of the envelope. So between them, or the space between your inner and outer, okay, membrane is what we call as your perinuclear space. Okay, that's the perinuclear space. And if you can see guys, no, there is this portion where your inner and outer membrane meet. Okay, they meet together to form this space. Okay, so yung space dito na nakikita nyo, okay, this forms now, or these spaces form now yung mga tinatawag naman nating nuclear pores. Okay? So, the nuclear pores. Okay? So, and then we can also see here the presence of your G. This is your pars granulosa. This is your nucleolus. This area here. So it has no the following components here. The more dense, okay, area, we have the pars granulosa. So it's just described as something that is granular. Okay, and this one naman is fibrillar. Okay, or fibrous. Okay, this is your pars fibrosa. So mamaya let's discuss no what's the difference, okay, of these areas, okay, or what are these areas, okay, found inside the nucleolus. Okay, so that's, or those are the basic structures of the nucleus or cell nucleus. Again, we have of course the nuclear envelope, okay, and the nuclear envelope has inner and outer membranes. Okay, and between, or these inner and outer membranes are separated by what we call your perinuclear space. And at some points, okay, itong ating inner and outer membranes, nagmi-meet po sila. And that forms now yung ating nuclear pore. Okay? And of course, if you can see guys, no, may dark yellow areas dito. Kung mapapansin nyo, may makapal na ano dito. This is what we call as your nuclear lamina, or also known as your fibrous lamina. Okay? It supports now yung ating nuclear envelope. And attached din dito sa ating nuclear lamina are your chromatins din. We have the heterochromatins. So kung nakikita nyo may lighter yellow, okay, so these are your heterochromatins attached to your lamina. But in areas kung saan meron yung mga nuclear pore, wala tayo makikitang heterochromatin dyan sa area na yon. So, kasi clear siya. Okay, it's a pathway, okay, for materials coming in and going out, okay, from that of your nucleolus, okay, and into your nucleus. Okay, so next. Okay. So guys, no, let's look into the clinical significance, okay, of our nucleus evaluation. Okay. So the simple microscopic evaluation of the nucleus provides no a great deal of information about cell well-being. Okay, so our pathologists, when they do examination of the cell structure, okay, the information that they get from the appearance, okay, of the nucleus is very vital, okay, in determining now the specific, okay, abnormalities or disease, okay, a patient may actually, okay, have. So this is important, I should say, okay, in giving right diagnosis dun sa sakit ng pasyente natin. Okay, by evaluating the nuclear size, evaluating the shape, as well as the structure, okay, of the nucleus. Because sabi nga natin, no, the nucleus, okay, plays a very important role in metabolism and in growth. So, and this is, of course, through the function of its individual structures. So, when these structures are abnormal, okay, or when these structures, okay, were damaged, okay, of course, that will result, okay, uh, to malfunction or abnormalities din sa buong cell natin. Okay? So, the nuclear alterations in dying cells are described as the following. We have your karyolysis. Okay, so when you say karyolysis, it's the disappearance of the nuclei, and this is due to the dissolution, okay, of the DNA, and that is because of an abnormal increase in the activity of our DNase or your deoxyribonuclease. Okay? So next, 'di ba, kung maalala nyo in histology and its method, in the laboratory, we use your DNase to extract your DNA. So in karyolysis, it's the one naman, okay, in the cell that causes dissolution, especially when it has an increase in activity. Okay? So next, pycnosis. Okay? Pycnosis, it's the condensation of the chromatin that leads, okay, to the shrinkage of the nuclei. Okay? So our nuclei, okay, becomes now, or the nuclei, okay, of the cells appear as dense basophilic masses. Okay? So parang ano ba 'yun? Pagkupit, parang kumukunat yung itsura, parang lumiliit, nag-shrink, okay, yung itsura nila, okay? And then lastly, we have your karyorrhexis, okay, or the fragmentation of the nuclei. Okay, so these are the different changes, okay, in the cell nucleus, okay, when the cell, okay, is dying. Okay, and this is very important in disease diagnosis, especially no in tumor or cancer diagnosis, okay, among patients. So first, okay, let's discuss about the nuclear envelope. So guys, no, just like the cell membrane, okay, your nuclear envelope or the nuclear membrane, okay, is a selectively permeable membrane, okay? And your nuclear membrane, guys, no, is 7 times thicker, okay, than the plasma membrane, and it's perforated. Okay, ibig sabihin meron siyang mga butas-butas, and we call these holes, okay, dun sa ating membrane as your nuclear pores. Okay? So, if you can see yung image kanina na pinakita ko sa inyo under your electron microscope, 'di ba? The nucleus or the nuclear envelope, I should say, has its outer nuclear membrane and the inner nuclear membrane. Okay? So, the outer nuclear membrane, kung mapapansin nyo guys, sabi ko kanina dun sa picture natin na nauna, it's continuous with your rough endoplasmic reticulum. And just like your rough endoplasmic reticulum, it's also a rough, okay, dun sa kanyang surface. Why? Because of the attached polyribosomes. Okay? So guys, no, we also have here the inner nuclear membrane. Okay? So the inner nuclear membrane is supported again by a rigid network of intermediate filaments. So yung pinakita ko kanina sa inyo, okay? So we call this as your fibrous lamina or yung nuclear lamina natin. Okay? So guys, no, yung ating nuclear lamina, it contains laminin receptors or lamin receptors, okay? And other lamina associated proteins, okay? And the purpose of these receptors and proteins is that they bind, okay, dun sa mga chromosomes natin and secure the attachment of these doon sa ating nuclear lamina. Okay? So, ipapakita ko sa inyo itong mga structures na to. So, if you can see it here, ito sila guys, yung mga pinagsabi natin. So, again, we have here the, how you call this one, the inner nuclear membrane, the outer nuclear membrane, and the space here is what we call as your perinuclear space. Okay? Beneath the inner nuclear membrane, itong dark area dito, okay, yan, this is your fibrous lamina. Okay? So, or your nuclear lamina. So, it's a rigid structure that supports now also, okay, this envelope here. And of course, attach now the heterochromatin, okay, dun sa ating nuclear envelope. Okay? So, guys, no, sabi natin, it contains the proteins, okay? So, what will happen if nagkaroon tayo ng problema, for example, with our nuclear lamina? Okay, so the clinical significance of that we have here. But before that, let's discuss muna, okay, yung nuclear lamina ninyo. Okay, so nuclear lamina, okay, is formed from a class of intermediate proteins. So, ito yung pinakamalaki blow up image nung image natin kanina. So, if you can see here, this is out the inner membrane, ito siya, and then we have the outer membrane. Etong gitna nito, this one here, this is the perinuclear space. Okay? So guys, nag-close sila dito sa area na to because we have here your nuclear pore. Okay? And beneath, okay, our inner membrane, itong green guys, okay, this is your nuclear lamina or your fibrous lamina. Okay? And it provides attachment dun sa ating chromatin sa loob ng nucleus natin. Okay? So when the nuclear envelope disperses of cell division, at least some laminin proteins, okay, remain attached to the membrane fragments, okay, and reassemble of the nuclear lamina immediately after cell division. 'Di ba? During cell division, nagkakaroon tayo ng restructuration, how do you call this one? Sorry. The restructure, okay, of your nuclear envelope. And after that, okay, cell division, there will be reformation that will happen. Okay? So, hindi nagkakaroon ng gulo yung mga chromatins natin because they are actually attached to this laminin or lamina. Okay? So, what will happen if nagkaroon tayo ng problem, okay, with our nuclear lamina? So, guys, no, titignan natin siya dito, the clinical significance, okay, of your, how do you call this one, the nuclear lamina. Okay? So we have what we call as your Emery-Dreifuss muscular dystrophy. Okay? So guys, no, in patients, okay, na meron nito, nagkakaroon sila ng mga muscle weakness, muscle wasting disease. Other than that, nagkakaroon ng contractures yung kanilang major tendons. Ano ibig sabihin? Yung itsura na parang nagkilo-kilo yung mga extremities nila, yung sa may paa, sa ankle, mga ganyan nila, or sa elbow nila. Okay? So, we have here a picture. Okay? So, makikita nyo siya dyan. Okay? So, ganun yung nangyari sa kanila. And the reason bakit nagkaganito, okay, yung ating pasyente is the problem actually lies, okay, dun sa problema or impairment nung nuclear lamina architecture. Okay? So, it's a genetic disease, okay, and this is an example of a laminopathy. Okay? So, anong mutation? So, saan nagkaroon ng mutation? It happened in your lamin AC. Okay? So, it causes now a tissue-specific disease. Okay? So, your Emery-Dreifuss, okay, is a genetic disease, okay, where there's mutation in the lamin AC. Okay? And these actually now cause problems, okay, dun sa striated muscle, adipose tissue, peripheral nerve or skeletal development, and of course, premature aging. Okay? So bakit? Kasi nagkakaroon tayo ng problema doon sa mismong structure as well, of course, the attachment nung ating mga chromatin. Kasi sabi nga natin, 'di ba, yung nuclear lamina nyo, it's very important in the attachment, okay, nung ating mga chromatin. So, it plays a vital role, okay, kapag nagkakaroon tayo ng restructure and reformation ng nucleus natin. But because of mutation, okay, it leads to abnormalities, okay, dun sa ating pasyente, lalong-lalo na dun sa kanyang striated muscle, adipose tissue, or in skeletal development. Okay? So, you have there the picture, or you have seen the picture, okay, of a patient with Emery-Dreifuss, okay, muscular dystrophy. And then next, we have the chromatin, okay? So guys, no, yung chromatin natin has two types, okay? So in non-dividing cells, it consists of the DNA and of course its attendant protein, okay, in a largely uncoiled state. Okay? So guys, no, yung ating chromatin has two types, okay? We have the heterochromatin. Under your electron microscope, kung nakita nyo kanina yung picture natin, di ba, it's coarse electron dense. Okay? Dark areas sila, black areas sila under your transmission electron microscope. Okay? In the light microscope, pag in-stain po natin sila, they are actually basophilic clamps. Okay? Guys, bakit basophilic? Kasi nga, they actually, how do you call this one? Um, absorb stains that are how you call this one, that are positively charged, basically because they are negatively charged structures. Okay, so bar bodies, okay, is an example of that. So your bar bodies are actually dense mass of heterochromatin found in females. Okay, so I do not know if you have heard, no, yung kwento ng isang athlete na napakagaling kasi niya. So they were like questioning, okay, kung ano ba talaga siya, baka lalaki siya talaga, or hindi siya oh, yeah, hindi nga siya babae kasi baka nga daw lalaki siya. Okay? So they actually found out na babae talaga siya, okay? Because they were able to see, okay, these Burr bodies, okay, sa kanyang cells, especially in leukocytes. Okay? So next, we have your euchromatin. Okay, your euchromatin, okay, as observed in electron microscope, it's finely dispersed and granular material. Okay? And dahil dispersed siya, guys, it's also basophilic pero lightly stained lang siya. Unlike, guys, no, dun sa ating heterochromatin, bakit? Kasi they are dense masses kasi, okay, of your chromatin. Kaya darker kasi compact sila, okay? So ulitin natin ha, saan yung mga sinasabi natin? So ito sila, guys. Ayan. So we have here yung mga dark areas na to, okay? Ito, yan. Mga dark areas na to, these are your heterochromatin. So kung makikita nyo, this is your TEM image. They are electron dense. Okay, right? Okay? This one naman, yung mga nandito sa paligid niya, yung mga gray, gray, gray materials na light dyan. Lahat yan. Okay, yung mga yan. Okay? Yung mga yan. Okay? Those naman are your euchromatins. So if you can see, they are dispersed, okay, sa paligid ng ating mga heterochromatins. And they are, of course, less dense, okay, or less electron dense than that of your heterochromatin. Okay, so again, dahil sila ay dispersed, sila ay lightly basophilic. Unlike your heterochromatins, they are darkly basophilic. Okay? So, next. Yan. Okay? So, guys, no, saan natin to nakikita? Guys, no, yung ating heterochromatin, okay, they usually are predominant, okay, dun sa mga metabolically inactive cells natin. Ibig sabihin, again, metabolically inactive cells. Okay? Those cells that are not undergoing um, active or are not active, okay, in cell cycle, for example. Okay? So, example of those are your lymphocytes, okay? O kaya yung mga sperm cells natin. Okay? So, or example, cells that produce products like your antibody, yung ating plasma cells. Okay? So, again, your heterochromatin, okay, are actually seen or are usually, okay, seen among cells, okay, that are metabolically inactive. Okay, inactive. Example, guys, we have your lymphocytes, the sperm cells, okay, and then we also have yung mga antibody producing cells natin or yung plasma cells natin. Okay? And then for your euchromatin naman, we have the finely dispersed granular material. So guys, no, naka-stretch. Okay, your euchromatin is an active chromatin. Ulitin ko, your euchromatin is an active chromatin. It's stretched out, okay? Why? So that the genetic information, okay, in the DNA can be read and transcribed. So, ibig sabihin, yung euchromatin ninyo, guys, it's ready for reading and transcription. Unlike your heterochromatin, dense sila, compact, hindi siya nakastretch. Kaya it's not prepared or it's not how you call this one destined, okay, for reading and transcription. That's for your heterochromatin. Pero dahil yung euchromatin ninyo finely dispersed, naka-stretch sila, okay, ibig sabihin the surface are exposed, okay, for reading and transcription. So ganito kasi yung tsura. 'Di ba yung heterochromatin nyo parang ganito yung tsura nila? Sorry, mahirap sumulat kasi dito. Yan. So, parang kulot-kulot. Yan. Yan. So, compact sila, okay? Hindi naka-expose yung surface dahil compact nga siya, okay? However, in your euchromatin, they are stretched. Okay? Dispersed sila. So, yung surface, or ganito, okay? Yung surface nila exposed, ready for transcription. Then, your euchromatin, okay, are active chromatin. Okay, they are stretched. Okay, and their genetic information in the DNA are ready for reading and transcription. Okay, so guys, no, your euchromatin, they are common, okay, or they are prominent in metabolically active cells. Yung heterochromatin nyo kanina, sabi ko, they are prominent in metabolically inactive cells, 'di ba? Yung heterochromatin. Ito naman, metabolically active cells. Example of these cells, we have guys yung neurons ninyo, your liver cells. Okay, 'di ba? Yung liver cells natin, they are very busy to metabolize yung mga pagkain na kinakain natin, yung drugs na tinitake natin. Okay? So, uh, they are very busy doing all of this function, okay? And of course, these active cells now, okay, has or these active cells, I should say, have no predominantly or the predominant type, okay, I should say, of chromatin in them, okay, is the euchromatin type, okay? So that's for your heterochromatin and your euchromatin. Okay. So guys, no, again, we have here the comparison. Cells with lightly stained nuclei are more active in protein synthesis. Ano na nga yung lightly, basophilic? Di ba yung lightly is yung euchromatin ninyo? Eh saan nga natin nakikita yung euchromatin? Sa mga metabolically active cells. Okay, kaya again, they are lightly stained and are more active in protein synthesis. And this, okay, or this refers now to your euchromatin. Okay? In light-chain nuclei with much euchromatin and few heterochromatic clumps, more DNA surface is available for transcription. Bakit? Kasi they are highly dispersed. Ibig sabihin, stretch, okay, yung ating structure, yung kanyang surface are ready for reading and transcription. Unlike your euchromatin, 'di ba, na masyadong clamp. Sorry, mahirap mag-ano guys dito sa yan. Kaya unlike ito, yung heterochromatin mo, ganyan siya stretch. Ito, kumpul sila. Okay? So, in dark stained nuclei rich in highly condensed heterochromatin, tightly called DNA is less accessible for transcription. So, ito siya. Okay? Again, pag dark stained siya, yan usually yung mga metabolically inactive cells ninyo. Like yung sinabi natin kanina, yung mga sperm cells natin, yung plasma cells. Okay, so they are metabolically inactive. And the predominant type of chromatin you see in them is yung ating heterochromatin. Okay? Okay. So guys, now this is just very short, okay, discussion. Okay, so guys, now we have here the smallest units, okay, or the smallest unit of a chromatin structure. Okay, so these are actually, or this structure is actually a complex of DNA and your histones. Okay, so kung makikita nyo, para siyang beads, di ba? Kaya nga itong structure na to guys, no, ay tinatawag nating beads on strings. Okay, ano ba yung mga structure na to? So if you can see, this is actually a nucleosome. Okay, so guys, yung ating chromatin, okay, is made up of chains of nucleosomes. Okay? So kung makikita nyo, this bilog-bilog here, okay, itong mga bilog-bilog na to, parang mga beads, these are your histones. Okay? And of course, we have different types of histones. Okay? So kung makikita nyo dito, we have two H2B, H3, and H4 histones. Okay? And dito sila. Okay? So, these are the histones. Itong mga nandito. At kung makikita nyo guys, yung histones natin, okay, they're actually, okay, surrounded by the central DNA. Okay? Andyan sila. Ito. Okay? And then, binding now this structure here, this helix, okay, dito, is yung tinatawag nating H1 stone. Okay? Or histone, I should say H1. Sorry, nawawala na ako sa ano ko. Okay, this is the H1 histone. Okay? So, guys, no, your H1, okay, with when the H1, okay, loosens, okay, nagkakaroon tayo ng unwrapping ng DNA natin. Okay, and this usually occur at least once every second, okay, to allow other proteins, okay, to join during the transcription. Okay? So, paano itong structure na to? Saan ito? Okay, dun sa ating chromatin. So, wait lang ah. Okay, so ito siya. Okay, so guys, no, if you can see here, okay, this is the packaging of chromosome into your chromosomal structure. So kung makikita nyo, these are actually blow-up image. This is your chromatin. Okay, and this view is under 1,400 nanometer. So guys, kukuha daw tayo ng konting portion nito, i-view daw natin siya. At 700 nanometer, itong portion na ito, ganito daw siya ang itsura niya. Okay? So, kung makikita nyo, itong area na to, na densely packed, this is the heterochromatin. And in this area is your euchromatin. Okay? And then at 300 nanometer, we have to look into this structure. This is your euchromatin. So, kung makikita nyo, ito yung kanyang itsura, guys. Okay? This is your euchromatin. Okay? So, the chromatin fiber with loops of chromatin fibril anchored into chromosome scaffold. Okay? So this is at 300 nanometer. And the blow up, okay, image, you can see here, this one, eto siya, yung portion na to, kinuha dito sa portion na to, pinalaki natin siya. This is as small as 30 nanometer, pinalaki natin siya. So we can see here the 30 nanometer chromatin fibril. So kung makikita nyo, ito na siya guys, yung structure natin kanina, di ba? Yung DNA and then the histones, yung mga beads natin. And at 11 nanometer, we're going to get this portion. Ito yung magiging itsura niya guys. 'Di ba? Ito na yung description niya, beads on a string chromatin. Okay? And if then we're going to get, okay, this portion here, okay, the blow up image is this one. Okay, this is an unfolded DNA double helix molecule. Okay, this is a view, okay, or this is actually a structure as small as 2 nanometer. Okay? So, ganito po yung itsura or pinakamaliit, okay, na structure na nakikita natin or nilalaman nitong or bumubuo dun sa ating chromosome. Okay? So, in dividing cells, chromatin is condensed and organized into discrete bodies called our chromosomes. And of course, the chromosomes, okay, are actually formed, okay, by two chromatids. So, ito yung single chromatid, okay, and another chromatid, okay? So, guys, no, the center, okay, of these chromatids, okay, is what we call as your centromere. So, they are actually joined by the centromere, the individual chromatids, okay? So, next, okay. So, next, we have this structure, no, in our chromosome. Okay, and this is what we call your telomere. Okay, and they are usually located at the end. Okay, nung ating chromosome. So, ganito yung itsura niya, guys. Okay, so if this is the chromosome, sorry sa drawing ko kasi mahirap guys mag-drawing dito. Okay? So, yung ating telomere, andito sila sa end portion, okay, nung ating chromosome. So, they're like shoe cap. 'Di ba? Alam niyo yung shoelace ninyo? 'Di ba yung shoelace nyo, guys, meron siyang parang plastic sa dulo. And the purpose of that is to protect yung shoelace para hindi 'di ba kasi yung shoelace ninyo parang nabuhol-buhol na thread yon. So, para hindi sila magiwaiwalay, nilalagyan nila yung plastic sa dulo. Yun yung shoelace cap, okay? And that's uh, para hindi nga masira, okay, yung shoelace. Ganun din sa chromosome. Para hindi magkaroon ng frayed chromosome, yung telomere andyan, okay, to protect, okay, yung ating chromosome. Okay? So, the telomere, no, sabi, it shortens, okay, with each cell division. Okay? So, studies indicate that telomere link is very important, okay, in knowing now, okay, the lifespan of a cell. So, the longer daw the telomere, the how you call this one, the longer, okay, the lifespan of a cell. Actually, there are studies, okay, or research conducted, okay, that um, found out, no, na itong telomere natin can actually be lengthened, okay, para magkaroon tayo ng, how you call this one, to um, reverse, okay, the aging process. Okay, turning back, okay, the clock of the aging process. So, yun yung research na kinandak nila. Okay, para daw i-lengthen, okay, yung lifespan. And of course, turn back, okay, yung tinatawag nating aging process. Okay, so cells must activate a mechanisms that maintains telomere to length, okay, or to lengthen, okay, and become immortalized. Okay, so a very good example, no, in cells that have been transformed into malignant cells, di ba? Yung mga malignant cells natin, guys, they are actually how do you call this one? Proliferating cells, okay? They duplicate fast, okay? Bakit? Kasi guys, no, these cells daw, no, have an enzyme called telomerase, okay? So this telomerase now is present, okay? And adds now repeated nucleotide sequence to the telomere end, okay, of these malignant cells. Kaya immortalized, okay? Kaya this is of course the reason na kung maalala nyo, 'di ba, sa ating how do you call this one, production ng monoclonal antibodies. 'Di ba, we have there the fusion of your plasma cells and that of your myeloma cells, which is a malignant cell. And the purpose of using is because to immortalize, 'di ba, yung antibody producing cell natin na tinatawag ngayon nating hybridoma. Okay? So yun yung purpose, okay? And yan ay characteristic po ng isang malignant cell, ng myeloma cell natin. Okay? So bakit? Ito daw yung reason, okay? Because the malignant cells, no, have this telomerase, okay, that enables them, okay, to how do you call this one? To add now repeated nucleotide sequence to the telomere ends. Okay? And these telomere ends, no, nung malignant cells, nalilengthen. Thus, no, how you call this one? Lengthening din, okay, yung kanilang lifespan, making them immortalized. Okay? So, yun yung clinical significance nung telomere sa ating chromosome. Okay? So, guys, no, this is just a brief, okay, introduction or overview, okay, nung napag-aralan ninyo sa isa nyo pang subject. Okay, so we have the human somatic cells. It contains 46 chromosomes. Ibig sabihin, we have 23 homologous pairs. Okay, so guys, in that 23, okay, you know naman, we have 22 pairs. Okay, and these are your identical chromosomes. Okay, so each chromosome of the pair contains the portion or same portion of the genome, and these identical chromosomes are called your autosomes. Okay? And the 23rd pair of the chromosome, okay, is actually your sex chromosomes. Okay? So yung 23rd pair ng chromosome natin, or na chromosome natin ay yung sex chromosomes natin. Okay? So guys, no, the chromosomal number found in most of the somatic cells of our body is called your diploid or your 2N number. Okay? So guys, you will know more of this cytogenetics ninyo when you'll be discussing about your cell cycle, meiosis, mitosis. Okay? So per egg cell and sperm, they have only 23 chromosomes. Tama ba? Okay, so haploid. Okay, number ng chromosomes sa egg cell, tapos ganun din sa sperm cell. Okay, as well as, of course, haploid amount of DNA. Okay, so the somatic chromosome number are reestablished at fertilization by the fusion of your sperm nucleus with your egg nucleus. Thus, we have your 2N or yung diploid ninyo. Okay? So, ayan. That's only an overview. Okay? Kasi hindi natin po ito sakop. Okay? This is for your other subject. Okay? So guys, no, next we have your nucleolus. Okay? So adding yung nucleolus natin, it's actually the structure, okay, at the middle of your nucleolemma. Okay? So parang yung cytoplasmic portion nung nucleus ninyo. Ito, nasa paligid or nasa gitna nito yung nucleolus ninyo. Okay? It's a spherical, highly basophilic subdomain of nuclei in cells and is very active in making proteins. So it's metabolically active. It is highly basophilic. Okay? It is a non-membranous region, okay, of the nucleus that surrounds transcriptionally active rRNA genes or ribosomal RNA genes. Okay? Non-membranous. Unlike yung mismong nucleus natin, di ba guys, meron siyang nuclear membrane or nuclear envelope. Ito, wala pong membrane that surrounds it. Kaya siya tinawag na non-membranous region. So nothing separates it from the rest of the nucleus. Okay, it's the primary site of ribosomal production and assembly. Okay, it's the site of ribosomal production and assembly. Okay, hold on. Okay, so the nucleolus, I should say, is the site of ribosomal RNA synthesis and initial ribosomal assembly. Okay, bakit initial guys? Kasi yung final assembly po, nangyayari po yan sa cytoplasm, so outside the nucleus. Okay, so your nucleolus varies in size but is particularly well developed in cells that are active in protein synthesis. Okay, so some cells may contain more than one, okay, nucleolus. Okay? Again, some cells may contain more than one nucleolus. Okay? So guys, no, yung dito sa site of ribosomal RNA natin, the ribosomal protein, okay, are actually synthesized in the cytoplasm. And then after that, they are transferred, okay, dun sa ating nucleus for sub-assembly. Okay, dun sa nucleolus. Again, sub-assembly or initial assembly lang. The final assembly, okay, of the subunits occur doon sa ating cytoplasm. Ibig sabihin, outside the nucleus. So, lumalabas yung mga materials doon sa ating nuclear core. Okay? From the nucleus outside, okay, papunta sa cytoplasm for the final assembly. Okay? So, guys, now we have the three morphologically distinct regions of the nucleolus. We have the fibrillar center, fibrillar material or your pars fibrosa, and granular material or your pars fibrosa. Okay? So guys, no, andito yung picture niya. Nasa image din kanina to. So if you can see here, the G here is your granulosa. Okay? This is the fibrillar center. Yung mga nasa gitna po nung ating pars granulosa as pars fibrosa natin. Ito yung mga parang puti-puti na areas, guys. Okay? This one, fibrillar area, itong medyo dark, yan siya. That's your pars fibrosa. Okay? Dito naman, mga granular areas, that's your pars granulosa. So guys, no, your fibrillar material or pars fibrosa, okay, these areas here, okay, represents now the site of initial ribosomal assembly. Okay? And they contain densely packed pre-ribosomal particles. So, ibig sabihin, ito yung site kung saan po nagko-contain nung mga, how you call this one, precursors for ribosomal formation na gagawin doon sa, or final assembly na gagawin doon sa cytoplasm natin. Okay? While this one, in your pars granulosa, okay, it contains now, sorry, the pars granulosa pala yung guys, yung sinabi ko kanina, okay? It contains now yung mga pre-packed, okay, or densely packed materials, okay, for the pre-ribosomal particles, okay? And then yung itong fibrillar material natin or pars fibrosa, it contains the genes, okay, that are actively undergoing transcription, okay, and large amount of RNA. Okay, so that's your pars fibrosa and pars granulosa. Okay? So, ano pa ang dapat nating alamin dito? Okay? So, guys, no, saan pa ba yun? Yan, the fibrillar center contains DNA loops of 5 different chromosomes. We have 13, 14, 15, 21, and 22. Okay? So they contain the rRNA genes, RNA polymerase, and the transcription factors. So these are the structures of your nucleolus. Okay? So this is actually a nucleolus from a nerve cell. Okay? So, yan. Okay. So those are the different structures, guys, no, of the nucleus. Okay? So again, we have there the nuclear envelope. Okay, anong tatandaan natin kay nuclear envelope? We have the inner and the outer membrane. And then at their middle, we have the perinuclear space. Okay? In the areas where your nuclear inner and your outer nuclear membrane meets, okay, form now yung tinatawag nating nuclear pores. Okay? And then beneath your inner membrane, we have your, ano ba yung di mo kakalimutan doon? We have there your nuclear lamina or fibrous lamina. Okay? That attaches now or that uh, holds now yung ating chromatin. Okay? And then we also have there the chromatin. Know the difference between your heterochromatin, guys, no, and your euchromatin. Ano na nga ulit? When we say heterochromatin, okay, we're referring to the coarse, okay, electron dense material. Ito siya, coarse, okay, electron dense material. And your euchromatin, okay, sorry, your heterochromatin, okay, ayan siya, the coarse ones, okay, electron dense, yung mga dark areas natin, okay? At saan sila nakikita, guys? They are predominant in inactive cells, okay? While your heterochromatin, guys, your euchromatin, I should say, itong mga finely dispersed chromatin dito, these are actually stretched chromatins ready for reading and transcription. At saan natin ito nakikita? They are usually seen or predominant dun sa mga cells that are metabolically active. Okay, those are your euchromatins. Okay, again, predominant in cells that are metabolically active like neurons and liver cells. Okay? So what else? The nucleolus, okay? The three areas, okay, found in your nucleolus. We have the fibrillar centers, the fibrillar material or pars fibrosa, okay? And then the granular material that contains now the precursors for the ribosomal assembly, okay? That will happen dun sa ating cytoplasm, final ribosomal assembly. Okay? So next we have cell death. Okay? So guys, no, in humans as in all other multicellular organisms, the rate of cell proliferation and cell death determine the net cell production. For example, in disorders that there is no increased cell accumulation, mas mataas yung rate ng cell proliferation or pagdami ng cells kesa sa namamatay via apoptosis or yung tinatawag nating program cell death. Okay? So uh, what conditions, okay, actually has this, okay, disorder? We have, for example, or I mean, what conditions, okay, actually formed. okay, or conditions that may have resulted from cell accumulation. Example, we have your hyperplasia, okay, and then we have cancer or yung autoimmune diseases natin. Okay? So anong ibig sabihin? Nagkaroon, example na lang sa cancer, 'di ba? There is increased cell proliferation. Kaya it will form what? Yung tumor or yung bukol na nararamdaman natin. Okay? So yung mga cancer cells natin, ano nangyari? Nagkaroon ng um, problema, okay, dun sa normal, okay, na apoptosis na nangyayari, hindi nangangamatay yung cell, instead dumadami sila, yung mga abnormal cells, okay? At yun ay characteristic po ng mga cancer cells natin, okay? And for disorders of cell loss, example, atrophy, okay? Atrophy of disuse, okay? Degenerative diseases, okay? AIDS and ischemic injury. So example sa AIDS, guys, later pag nasa mycology and virology na kayo, 'di ba, yung patient kasi na may HIV infection, hindi naman kaagad siya magkakaroon ng AIDS, okay? Magkakaroon lang siya ng AIDS kapag nagkaroon na ng decreased number of your CD4 cells, okay? CD4 T cells. Yun kasi guys 'no, yung at ina-attack po nung ating mga HIV, or how do you call this one, the HIV or the virus, okay, that's causing the HIV infection. So it attacks those cells, and when these cells now, okay, level sobrang baba na, that's the time no na magkakaroon tayo ng, or yung pasyente ng increased susceptibility dun sa infections na, especially yung mga tinatawag nating opportunistic infections, okay? Those infection that do not usually, or microorganisms I should say, that do not usually cause infection sa healthy normal individual, pero since impaired na yung kanilang immune system, okay, nagkakaroon sila ng sakit caused by these opportunistic microorganisms. Example, infection with your Candida albicans. Okay, nagkakaroon sila ng oral thrush. Okay, what else? Infection with rare types of cancer like Kaposi sarcoma. So, mga ganun. Okay? So, disorders of cell loss. Okay? So, guys, no, there should be balance, okay, or homeostasis between cell division and cell death. Why? Kasi pagka there's increased cell loss, of course, that will result to disease, okay, due to cell loss. Okay? Like AIDS, your Alzheimer's, your Parkinson's, di ba? That lead to what? Damage, okay, of the, how do you call this one? The nervous system cells, okay? A plastic anemia, okay? Problems with your blood cells. And then myocardial infarction, okay? Nagkakaroon tayo ng damage sa cardiac muscle cells natin. Okay? And then we also have your cell accumulation disorders. Mas madami yung cell na nabubuo or nagpo-proliferate kesa sa cells na nag-undergo ng program cell death or yung apoptosis natin. Example, in patients with cancer, with autoimmune diseases like your lupus erythematosus, glomerulonephritis, o kaya naman viral infection. Okay? So these diseases here, I will not be discussing them kasi you will know more of them pagdating nyo sa higher subject, and you will understand what cell death and cell division. Okay? Kung bakit cell loss disorder sila. Example yung sa AIDS na lang. In-example ko siya sa inyo. Okay? Di ba? Nagkaroon ng AIDS yung pasyente because of the decreased number of CD4 cell. That's cell loss. Okay? Cell accumulation naman, for example, in cancer, di ba? Increased proliferation of cells. Pagdami. Mabilis na pagdami nila. Okay? Kesa sa nangangamatay. Okay? So, nagkakos ng accumulation. That leads, kaya nga yung ibang cancer, di ba, may bukol, okay, or tumor na tinatawag natin. Okay? So, guys, no, for the cell death, we have two major mechanisms. We have what we call as your necrosis. And when we say necrosis, it's something pathologic. Okay, bakit? Kasi meron tayong cell death na non-pathologic or physiologic. Tawag natin. Okay? So, necrosis or accidental cell death is a pathologic process. So this usually occur when cells are exposed to unfavorable physical or chemical environment that cause acute cellular injury and damage to the plasma membrane. Example, um, ano ba, necrosis example, nagkaroon ka ng sugat, okay? Nagkaroon ka ng, na-prick ka ng thorns, okay? Ng rose, ng Valentine that leads to infection or that led to an infection dun sa kamay mo, okay? So, nagkaroon ng infection sa kamay, yung area na infected, pwedeng mamatay, okay? Or may localized death ng cells doon because of the infection, okay? So, that's necrosis. Damage to the plasma membrane may also be initiated by viruses or proteins called perforins. Okay? So guys, no, tandaan ninyo, the characteristic feature of your necrosis is rapid cell swelling and lysis. Okay? Tandaan ninyo, in your necrosis, usually guys, nagkakaroon tayo ng damage ng cell membrane. In the other type of death, we have apoptosis. Hindi nagkakaroon ng damage dun sa cell membrane. Instead, nagkakaroon tayo ng bleb formation that lead to formations of yung pinatawag nating apoptotic bodies. Okay? So, mamaya may picture dyan, magpapakita ng difference nila. Okay? So, guys, no, apoptosis naman, okay, was referred to in the past as programmed cell death. So, your apoptosis is characterized by controlled autodigestion or tinatawag yung self-digestion. Okay? So, the cell dies with dignity without spilling its content and damaging its neighbors. Okay? So the cell is actually releasing yung kanilang the enzymes, lytic enzymes sa kanyang sarili lang, okay? Without releasing it to the neighboring cells. Okay? So ibig sabihin siya lang yung affected. So it dies with dignity without without how you call this one, causing damage sa neighboring cells niya. Okay? So, unlike in your necrosis, okay, that there's bursting, okay, and swelling, okay, that leads to the damage of the cell membrane. Okay, so next, we have here the morphologic and biochemical features of apoptosis. Hold on. Okay, we have DNA fragmentation. So it's an irreversible event that commits the cell to die. Okay, so a result of your calcium-dependent and magnesium-dependent activation of nuclear endonucleases. So these enzymes leads now to the fragmentation of the DNA. Okay? And then we have decrease in cell volume. So nagkakaroon po tayo ng shrinking ng cytoplasm. Bakit? Nagkaroon kasi ng damage dun sa cytoskeletal elements, okay, nung ating, how do you call this one? Nung ating cell. Di ba? If this is the cell, dun sa ating plasma membrane, nagsusupport dyan are your cytoskeleton or the cytoskeleton. So, nagkakaroon ng restructure niyan, okay? Kaya nagkakaroon ng reformation nung ating the plasma membrane, okay, leading to the shrinkage of the cytoplasm. Okay, ribosomes become clamped within the cytoplasm. Okay, and then your rough endoplasmic reticulum forms a series of concentric whorls. Okay, and most of the endocytotic vesicle fuse with the plasma membrane. Okay, so mga endocytotic uh, vesicle daw, they fuse with the plasma membrane and form bodies, okay, that we call as your apoptotic bodies. Okay? You will know more of the formation of your endocytotic vesicles, guys, pagdating po natin doon sa cell cytoplasm natin. Okay? The cell cytoplasm. And then after that, we have loss of mitochondrial function. Bakit? Nagkaroon tayo din ng changes dun sa mitochondrial membrane channels natin. Okay? And then finally, there's membrane blebbing. Okay, magkakaroon ng alteration dun sa ating plasma membrane and eventually will lead to the formation of apoptotic bodies. Okay, that results in cell breakage. So guys, no, I have here a picture. This is the last slide showing the difference between necrosis and your apoptosis. So guys, no, kung makikita ninyo, ayan siya. This is the cell. When it goes necrosis, nagkakaroon ng bursting. Nakikita nyo, nagkaroon ng pagsira doon sa plasma membrane. So, lumalabas yung laman ng cell. Okay? So, nagkakaroon ng injury sa cell membrane. Other than that, there's swelling. Merong inflammation. Okay? In your apoptosis, nagkakaroon tayo ng DNA fragmentation. What also happens? Nagkakaroon ng shrinkage sa cytoplasm due to the reorganization of the plasma membrane. Nakita nyo, nagkaroon tayo ng restructuring kasi nung cytoskeleton. Pero wala naman siyang butas. Unlike here, 'di ba, na nagkaroon tayo ng bursting, okay, ng nuclear membrane. Kaya nare-release, sorry, the plasma membrane, I should say. Kaya nagkakaroon ng burst out, okay, nung mga laman nung cell natin. Here, hindi. Okay? It dies with integrity, sabi niya nga. Okay? And then after that, it leads to membrane breakdown. Okay? And then eventually disintegration and inflammation. Okay? So here naman guys, after na nagkaroon tayo ng the structure ng kanyang cytoskeleton, nagkaroon nga ng shrinkage and eventually nagkaroon tayo ng formation ng endocytic vesicles or the how you call this one, the fusion of your endocytic vesicles dun sa ating plasma membrane. Parang kumbaga parang paano ba i-describe? Um parang nakakaano na ba kayo ng dough? 'Di ba yung isang malaking dough pag kinurot mo siya, ay magkaka-form ka ng maliliit na bilog. 'Pag kinurot mo siya, hilain mo, may mafo-form ka. So, parang gano'n yung dating niya, okay? So, ayan siya, naghiwa-hiwalay sila, okay? The endocytic vesicles now joins now yung plasma membrane tapos magkakaroon ng formation ng mga bilog-bilog, okay? And these are your apoptotic bodies, okay? Unlike here, guys, nagkaroon tayo ng burst okay, of the plasma membrane or of the cell, releasing its content to the neighboring cells. So, pwedeng ma-harm yung neighboring cells natin. But here, no, it dies, no, with dignity, kanya. Okay, kasi ang nangyayari, nagkakaroon lang tayo, no, self-digestion or autodigestion. Okay? So, that's necrosis versus apoptosis. Again, when we say necrosis, it's something pathologic. When we say apoptosis, it's, okay, also known as your programmed cell death. Okay, so it's usually of a natural occurrence. Okay? So, like for example, the shedding of the skin cells. Okay? So, yan. Itong necrosis, example, infection. Okay? So, natusok ka. Okay? That leads to pagnanana. Okay? Inflammation ng tissue natin. Okay? So, that's a result of necrosis. Okay? So guys, no, please do watch no the videos in the suggested self-directed activities that will help you understand more, okay, of these how do you call this one of the topic, okay, necrosis and apoptosis. Okay? So that's all. And no, I will not be giving you muna a quiz dito sa cell nucleus. Why? Because I will be combining the quiz dito with the quiz ng cell cytoplasm. Kasi masyadong maiksi po ito. Okay? Yung cell cytoplasm, dun na natin siya, pag na-discuss natin siya, dun natin i-combine yung quiz natin. Okay? So please do not forget to visit the suggested self-directed activity section and of course to read your books. Okay? So, don't forget also to access no yung ating application. Kung makikita nyo, the application is actually the whole cell. Okay? So, pagdating ninyo sa the cell cytoplasm next week, wala po tayong application doon kasi yung application po ni the cell nucleus at ni the cell cytoplasm ay isa na lang po. Okay? At dito siya naka-attach. Okay? Sa topic 2 po natin. Okay? That's all for this week and I hope you will give time again to listen para sa topic natin next week. Mahaba po yun at importante na pakinggan nyo. Okay? Thank you very much, guys.